Welcome to the third day of the webinar series, Health and Diseases Contemporary Concepts. Today we have Dr. Sunil Raghav from Institute of Life Sciences, Bhubaneswar, a scientist and Swami Fellow, to deliver an interesting talk on the contemporary issue of COVID-19. So, I welcome Dr. Sunil Raghav. Dr. Sunil Raghav got his PhD in 2007 in biotechnology from IGIB New Delhi, a young guy with a lot of accolades, and to encourage all the youngsters present over here with his work and the impressive talk he is going to deliver. He is a postdoctoral fellow from 2007 to 9 in Molecular Immunology Group at Turku Center for Biotechnology, Finland, as SIMO SITRA fellow. During his tenure, he established next-generation sequencing methods for functional genomic analysis to understand human T helper cell differentiation. This is like the next generation sequencing techniques were just born during this period, and there were no ready-made pipelines for this sample preparation and data analysis. So he is a pioneer in that particular method. He was also a postdoctoral scientist from 2019 to 2013 at Institute of Bioengineering at Lucerne, Switzerland. He used functional genomic approach to explore the role of co-registered proteins in fat cell differentiation and develop methods to perform genomic analysis of co-regulation in different biological systems. He is now a scientist and Ramanujan Fellow at the Department of Biotechnology, Institute of Life Sciences, Bhubaneswar. And the group's major goal is to understand the mechanism of response development in immune cells and thereby develop methods to modulate immune response in different diseases for therapeutic purposes. He has a lot of publications in peer-reviewed journals, but most importantly, he is involved in his COVID testing for the state of Orissa at Institute of Life Sciences. With these words, I welcome Dr. Sunil Raghav to deliver his talk. Dr. Raghav.
Thanks, Luna, for nice introduction. And first, I would like to thank Dr. Patro and all the academies, Academy of Science and Academy of Neuroscience, and ILS, because that is also one of the organizer. So thanks for giving me this opportunity to give this webinar and discuss about what we are doing on COVID-19 and then what type of research we are doing at ILS. Am I audible? Okay, so I will go ahead. I think it's fine. So good afternoon to all of you, that faculty, colleagues, and students that are attending this webinar. So before going into my main talk, I will just like to give you an overview or animation of this coronavirus, that is basically SARS-CoV-2, as you can see here in this slide. So this is coronavirus. We call this coronavirus because if you see this structure, animated structure, you can clearly see a crown-like structure. So what is this? Basically, that these red proteins present on the surface of this virus are basically spike proteins. We have heard of, definitely everybody have heard of the spike proteins. So these are the spike proteins, and then this gray layer is basically the lipid membrane layer that protects the genetic material of this virus that is present inside the core. And that is basically the RNA that is condensed and captured by the nucleocapsid protein inside. So this whole looks like a crown. That's why we call it a coronavirus. And this is the main reason or the main problem behind this COVID-19 pandemic. So coming to the main talk, I'm going to tell you about here in this webinar is the genetic and the immunological aspect of this COVID-19 infection. So I will tell you about the COVID-19 virus genome, how it looks like, and what is its impact when it infects the host cells or the humans. So here, if you look at this simple diagram of this coronavirus, you can see the same like a structure. There are these spike proteins, then you have this membrane, and this RNA is as the genetic material inside. And on the right side, you can see an electron microscopic image of the same virus, SARS-CoV-2, that causes COVID-19, and that is infecting a lung epithelial cell. So this interaction, that is basically the spike protein interacting with this, and once this interaction happens, the virus goes inside this host cell. So coming to like what is the adaptation if you have this RNA inside the virus, because there are different type of viruses. In SARS-CoV-2 we have RNA, but there are viruses which have DNA. So what is the difference between if you have RNA inside or the DNA inside? So if you see in this slide, there there are two major type of viruses that are basically RNA as the nucleic acid material or the genetic material or the DNA. So what happens if you have RNA? The adaptation is that RNA can be directly translated into proteins. That's the central dogma. Maybe you know that DNA is first transcribed into mRNA, messenger RNA, and the protein. But if you already have RNA as the genetic material, then there is a adaptation or the benefit to the virus that it can directly make the proteins inside the host cell by directly using the host machinery for making the proteins. Whereas in the DNA viruses, basically first the DNA has to be integrated in the host genome, and then RNA will be made, and then that will be used to generate more viruses inside. So in case of coronavirus, this COVID-19 virus, there is adaptation that it's already a positive strand RNA virus, which can directly make proteins inside host cells and generate more of the virus particles. So if in a simplified version, if you see that what type of genome architecture this SARS-CoV-2 virus has, so the total length of this RNA that it has is around 29 kilobases, 29,000 bases, you can say. It's nearly 30 kb length of this RNA. And this is a single stretch, which is basically poly A tail here at the end. And this leads to production of different proteins like this proteases, as you can see here. Then replicase. This replicase is basically the RDRP gene, RNA dependent RNA polymerase, that helps in the replication of this RNA inside the host cell. And then these proteins like a spike protein and all, which are formed when once this RNA is inside the host cell. So now the question comes, I think this is very interesting slide. Why this SARS-CoV-2 virus that is causing COVID-19 is more infectious as compared to SARS-CoV-2 that was known before in 2011 there was infection. So what is the difference between this SARS-CoV-2 and SARS-CoV, and why this is much more infectious? So as I told you, this spike protein, that is basically a trimeric protein, that is very important for interaction with the ACE2 receptor that is present on the human cell membrane or the host cell membrane. This is the ACE2 protein. So what happens in case of SARS-CoV, this receptor binding domain of this spike protein is always in an open configuration like this for binding with the human ACE2. And that's one of the reason also that there were high level of antibodies for SARS-CoV in the individuals when they got infection, because this domain was always in an open position and it was detected by the immune cells of the host. But in case of SARS-CoV-2, that is basically the COVID-19 virus, what happens? There is a important like a pre-activation by this furin and protease that cleaves it hinge region, and then only this protease goes up and interacts with the human ACE2. So that is quite nice adaptation in this virus because of some mutations, 5 amino acid mutations that happen in this spike protein that lead to this adaptation in this virus. And because of that, what happens? There is high affinity of this receptor binding domain of a spike protein of SARS-CoV-2 or COVID-19 with the host cell as compared to SARS-CoV. And there is a pre-activation by this furin or a TMPRSS2 protease. Maybe you have heard of that there is a ACE2 receptor, there is a TMPRSS2 protease present on the membrane of host cell, and that cleaves that hinge region, leading to this activation of this receptor binding domain. So these two things led to adaptation to SARS-CoV-2 for more infectivity as compared to SARS-CoV. So this is the same slide, just as I told you, that this virus basically binds to the ACE2 receptor present on the host membrane, host cells that are present in the thoracic cavity or the lung cells, lung epithelial cells. This ACE2 receptor is present. So what happens? This SARS-CoV-2 bind through the spike protein to this receptor ACE2, and then this genetic material comes out of the virus, and that is basically used, that uses the host translation machinery to produce its own proteins, a large number of different proteins like spike protein, nucleocapsid protein, and all these. And then these, during this process, it undergoes encapsulation again. It makes the more copies of the viruses, and then it comes out of the cell to infect more of the host cells, either in lungs or the thoracic cavity. Same was basically for the MERS-CoV virus or the SARS-CoV. The process is same, only the infectivity is more because it has more capacity to interact with the receptor ACE2 and with high affinity. So if you look at, if you track it, basically what type of different infections, and this is a very important slide in terms of why we see so much of difference and how, what is the percentage of onset of symptoms and symptomatic asymptomatic cases. So if you look into here, that during day 1 to 3, 80% of the cases are basically developing these mild symptoms only, and that comes during infection from day 1 to day 3. That you get fever, you get throat infection like a rough throat, and then some cough, and the virus is cleared during this early phase itself by the immune response. So there is an immune response element is strong, and then it doesn't go to this day 4 to 9, and already the virus infection getting cleared in by 1 week. But what happens if it doesn't get cleared? It starts going to the lungs, and that is the problem when there is more infectivity, or what you can say, the symptoms get severe. So you have more of lung involvement, the respiratory problem, so you will feel problem in respiration and all, and some eyes oxygen requirement might be there. And this is basically in 14% of the cases. During the late stages, when the lung get fibrosis, or the, what happens first, the virus infects the lung epithelial cells in the alveoli. But if the symptoms is not contained by the immune cells, then you get strong cytokine storm. You must be having heard of cytokine storm, and that leads to fibrosis of the lungs, and ultimately the infection reaches the blood. And during this stage, this is the latest stage, and ICU is required, ventilator is required, and the chances of recovery is really less if somebody reaches to this stage. But the percentage is much less in this case. What are the symptoms if you look into? So if you see the percentage, it clearly says that most of the cases you get basically fever, dry cough, and sore throat and all, and the virus get cleared. As I told you that 80% of the cases you have only mild symptoms. But when there is like a shortness of breath and respiration, it shows that the infection has reached to the lung, deeper into the lung, and then it starts basically the oxygen deprivation stage. And as you can see a below image, basically this is the CT scan of lungs, where you can see these white patches. So these white patches are basically fibrosis or the fluid accumulation in the alveoli of the lungs, where the oxygen exchange happens. And in this stage, the ICU, basically ventilator situation comes, and that is the problematic situation of severity. So overall, these are the three different stages. So stage 1 is basically early infection of the virus, and during this stage, there is lymphocytopenia, basically the decrease in white blood cells, and there is these markers, C-reactive protein, D-dimer, active dehydrogenase, these molecules go up. Then during the pulmonary phase, when the lung get infected, you have very high levels of these proteins. These are basically the liver proteins as well. These are acute phase response proteins that increases the inflammation. And in stage 3, you get hyperinflammation phase. That we call as cytokine storm, where is very strong host response. And this host response is basically lethal in the lungs, and that creates almost like fibrosis like situation. So, and IL-6 is the major cytokine that is known to link to this hyperinflammation phase. So now, after getting this introduction, I would like to tell you what we are doing for this COVID-19 in at ILS, our institute, because we are doing a large number of testing. We have already done around 90,000 tests. So at the same time, we are trying to understand what type of virus genome is there in the population that we have. So what we are doing this next generation sequencing of virus genomes. From March to June, we have already done. Then we are trying to understand the immune system of these COVID-19 infected patients. We are collecting the blood samples and swabs and different other samples to understand that. Then we are doing generating antibodies. Some groups are generating antibodies in our institute for this virus proteins for detection. Then we are doing some drug repurposing or drug screening for uh, this COVID-19 to identify if we can identify some of the composition. And we are also generating animal models as we have the BSL-3 facility, biosafety level 3 facility for handling such pathogens using hamster model and human ACE2 receptor transgenic mice. So I'm not going to discuss about all these different things. I'm going to discuss and tell you about what are the findings from us in terms of virus genome, what type of virus genomes are there that is in the Indian population, and what type of immune responses we are getting in COVID-19 patients in case of asymptomatic, symptomatic cases, and even the contacts. Why the contacts are not positive when we detect them. There is some preliminary observation, so I will tell you about that in a minute. So if you look at the virus genomics, this is a overview slide. Then I will tell you some of the results. So what are the major objective here for doing the virus genomics is to identify what are the different viral strains that are prevalent in Odisha population. But I would say this is not Odisha population because these are migrants to Odisha, but they are migrated from different places or different states in India, that is Maharashtra, then Kerala or Tamil Nadu or Hyderabad or Gujarat. So basically it's covering the whole Indian population. Then we wanted to see, is there any difference in the virus genome in asymptomatic and asymptomatic individuals when they get infected? So what is the current status of our analysis? So we have sequenced around 225 genomes from these different COVID-19 infected individuals. We have submitted the data to GIS in this online database also. And this work is done under PanDBT 1000 viral genome sequencing initiative. So we are doing it from ILS. Some of the samples we got, early samples were from RMRC Bhubaneswar, because initially we were not doing the sequencing in March start. So we took samples from RMRC and the other samples we have from ILS when we were doing testing. Now we have plans to do 96 more virus genomes because we have sequenced from March to June only. So now we are planning to sequence around 96 more genomes from June to September to see what type of new clades or new virus genomes are coming. So observations I will come now in the next slides. So if you look at this slide, so what type of virus clades or virus strains we see in the genomes that we have sequenced? If you look at this figure, first figure, we have most of the patients or the most of the infected cases from 20A, 20B, and 20C. And these are basically, if you look at right side picture, they are mostly basically from May to June. You see most of them have 20A and 20B cluster, okay? There 19B strain as well we see. 19A and 19B basically is named because they are the initial strains that came during the migration of individuals from European side and the South Asian side. And we have 20C also, but this strain is very less. Somehow this is not adapted to that much. And these news were covered by media as well. This is just the phylogenetic analysis of those 225 strains. So here, if you look at what we try to do, we try to associate these clades with symptomatic and asymptomatic cases. And if you look at this highlighted region, this yellow line shows that 20B clad is there. And if you see that red line along with, red indicates basically the symptomatic cases. So it appears that 20B clad is more associated with the symptomatic cases. So if you are infected with 20B strain, there are high chances that you will get some symptoms. It's not about severe and mild, because most probably these were mild cases, because we don't have that many details, because these samples came for testing. But this is very clear that 20B clade has more infectivity and they have more symptomatic cases in this. If you look into the variation level, you can see that 19A, 19B strain has quite different. So this is basically ball plot. The balls indicate the size of the ball, indicate the sample count, and the color indicates the clad. So you can see that the 20A, 20B clad had different variations at different genes. So S means spike gene here. You can see that the mutation is at different location as compared to these 19A and 19B. So it shows that the virus evolved with time and it gained different mutations. This is a very interesting figure. Basically, don't go into the details. This shows about the transmission dynamics of infection. So if you look into, there are two branches overall, and this is for the how the migration happened of the virus within India. So if you look at the lower branch, you see mostly the brown color, and this brown color goes to Gujarat. So it shows that there is one strain that mostly originated from Gujarat and the transmission happened from Gujarat. On the other side, we see that there is one more major hub, and you look into that this major hub is like from different regions. So it's basically Chennai, it's showing different regions origin, like there are some cases which are foreign as well. And then they migrated to different places of different states in India. But one major portion is coming from Gujarat. It's clearly showing. And it might be that we have a lot of individuals that migrated from Surat and Ahmedabad in Gujarat. Now, if you look into this transmission dynamics in terms of different countries as compared to India, so if you look here, you see that there are again two groups. One is from South Korea or South Asian countries. So it shows that one major transmission happened from South Korea, and the other major transmission happened from Italy and basically India we call, because this is from Gujarat they came. But this happens from European region. So Europe and South Asia. And the South Asian strain basically is 19B that we later on identified. That 19B came from South Asia, and 20A or 20B it came from European region, and that migrated to different states in India and evolved further. Now, what we looked into that these 20A, 20B, why they have more like adaptation, why they have more infectivity. So we found there is one mutation in a spike protein. This reason that I told you that is very important for this interaction of spike with the human host cell receptor ACE2. What we found that it has more of this G614 as compared to the D614 mutation. D is basically aspartic acid and G is basically glycine amino acid. So aspartic acid is changing to glycine. And if this change happens with time, as you can see that during the June phase, you have more of these strains with this mutation. And you look into the infectivity or the viral load, you see that the viral load goes up as shown by the decrease in this CT value or detection rate, because you can detect much more easily in the RT-PCR. So there is more of viral load. So when this mutation is there, more of viral load is there. And if the more viral load will be there, there is chances of more infection. What is the impact of this mutation? So if you look at, this is the region where I told you that the receptor interacts with this host cell um, receptor binding domain of the spike protein in virus interact with ACE2 receptor. So what happens when this mutation happens? This is the aspartic acid. So there is this hydrogen bonding. But when this receptor it changes, this amino acid is changed to glycine, this hydrogen bonding is lost. Because of this, what happens? That this is the TMPRSS2 that I was telling, that protease that cleaves this hinge region. So you see this, there is a distance between interaction site of protease with the spike protein. What happens when this mutation happens? This interaction site comes closer, and this interaction site, what it does, it leads to increased cleavage of this spike protein, leading to increased interaction of this RNA binding domain with the ACE2 receptor, and thereby leading to increased infectivity of the virus. We call it virus shedding, increased virus shedding. So this we have submitted these preprints, basically publications in bioarchive, open archive, and they are in review in other journals as well. There is one more interesting finding that during testing, what we did, we checked these virus genome in fecal samples of some of the individuals, and you can see here that there is, in fecal sample also, the virus genome can be detected. So it shows that definitely it might be that virus is shedded through fecus, and that came basically, so we asked the state government to look into those things also in the containment zones, so that the sewage disposal and sewage treatment should be in a proper way in the containment areas and in the quarantine centers, so that infection should not happen because of that. So this is the genetic part. Now we see that there is some difference between the symptomatic and asymptomatic cases. Now we wanted to see that what type of immune response happens when you have this SARS-CoV-2 infection. So what we did, we collected samples from Ashwini Hospital. These are basically COVID hospitals in Cuttack. Then we collected from KIMS, that is a KIMS hospital. Then there is one more hospital that is from Bahrampur, that is Tata Hospital. We collected some of the like blood samples, then urine samples, swab samples. The idea was to look into what type of immune response these individuals are developing. And one more interesting thing we did, we basically collected direct contact samples also from direct contacts that were not detected RT-PCR positive, and they didn't develop any symptom also, and they came for 4 days in the same bus along with these migrants. So we were surprised, like why they didn't develop this infection, and what happened, what might be the reason. So these are the different groups that we made, like asymptomatic, then symptomatic with mild flu-like illness, symptomatic cases with mild respiratory distress. So these are mild symptoms. Then symptomatic with severe respiratory distress. They were in ICU. These samples we got very few. Then contacts. These are basically direct contacts of RT-PCR positive patients, but they were not detected positive when they came for the test, and they had no symptoms at all. Then we took basically 2 time points for basically positive patients, and then when they started recovery, so basically 2 to 5 days, and then 8 to 10 days of the same patients. It's like a longitudinal sample, and then pure negative individuals to look into what is the difference in the immune profile. If you look into, so what does it mean by immune profile? So we first check the antibodies against COVID-19. So this is the ELISA. We use an ELISA technique to measure the antibodies. And these are the total antibodies. This can be IgG, IgA, IgM. So here what you see, these are the controls. You see that they don't have antibodies against this infection at all. Then we have this Ganjam. From Ganjam, we collected these samples. So these are PCR positive cases. And these are the contacts. Surprisingly, in contacts, what we see, that they have very high antibodies as compared to these PCR positive cases where a lot of individuals they don't develop antibodies strongly. We have in context very strong antibodies. When we look into what type of antibodies they have, we found that they have very strong IgA antibodies, and these IgA antibodies are subtype which is present mostly in the respiratory tract, basically in the mucus in our respiratory layer. This is heavily present, and this is well known according to literature that if somebody has very high antibodies in the respiratory tract, it can neutralize the viruses. So we just have to check now whether these individuals have the neutralizing power in these antibodies or not in their serum. So that we are going to check now. But it appears that they got some exposure in the start, but they have capability to develop very strong antibodies. So that's why they didn't came very positive in our RT-PCR, and they developed antibodies very fast. So they were not contracting the disease. If you look into these R1 and R2 samples that I told you, like these are 2 to 5 days after hospitalization, and then R2 is 8 to 10 days after hospitalization. What you see here, that a lot of individuals don't develop antibodies even after infection. Only some of the individuals develop antibodies, and this is the problem that I told you, that somehow the spike this is not very open, this receptor binding domain, and that is not very visible to the immune cells, and that's the reason that antibodies don't develop very strongly in some of the cases. And this might be a problem, hopefully it should not be, and it might be a problem in reinfection cases also if the antibodies don't develop very strongly. But as here you can see that a lot of individuals really develop very low amount of antibodies, and they have less of IgA. So, and these are very important for clearing the infection. So what we are doing now next also, to understand what type of this B cell and T cell receptor are present on these cases. Some of them are developing high antibodies, some of them are not developing. So what is the difference? Why their B cell and T cell are different? So we are trying to do some single cell experiments to understand that as well. Now coming to, so antibodies, we see that the contacts have more antibodies as compared to the PCR positive cases, and that might be the reason they have utilized it. Now we look into the cytokines in their blood serum. What type of cytokines they have in case of infected PCR positive versus the contacts. So if you look into here, that this is already reported that IP10 and MCP3 are severity indicators. If these proteins are very high in somebody, they can go into severe complication. So if you look into here, the Ganjam PCR positive cases and contacts, there is no much difference in IP10. Whereas after recovery, the IP10 goes down, and similarly MCP3 also goes down, and in contacts we have less of MCP3. So somehow these contacts already have less of these MCP3 and IP10, which can lead to severity of the disease. And during recovery also, these factors go down. But in our study, we found these 4 different molecules like MIF, macrophage inhibitory factor, IL-1 receptor alpha, these 2 different cytokines also. As you can see here, in context, they are very high as compared to the PCR positive cases. And this is quite interesting. It might be that these are protective cytokines or protective factors that are present in context. And in recovery phases also, you can see that these factors go up compared to early days of infection. So this shows that these factors can be protective factors, and they can be like a protective markers that can be used. So this we are still working on it. Now, I would like to conclude with 1 or 2 more slides. So what we are doing in terms of COVID testing in ILS, so we have done really a large number of samples till now. So from March, we have done around 90,000 samples. We started with 44 samples in around mid of April, and now we have around 90,000 samples tested out of total 260,000 tests done by basically total test done. And then CM also, like a state, appreciated much. These are some of the pictures how we do it in our DSL3 facility. So finally, I would like to thanks the Department of Biotechnology for giving us this opportunity to do all these work and funding and everything. Then government of Odisha, because government of Odisha really supported well, and then we did a lot of testing, so they have supported in all the aspects. Then ICMR, RMR days, and then providing the samples for COVID hospital, KIMS hospital. So these samples now we are using to do a lot of different, basically analysis in terms of single cell analysis, B and T cell analysis, and we are trying to understand what type of differences between sensitive and these asymptomatic, symptomatic, and severe cases. We are trying to understand in Indian population what's happening. And NIBMG is the coordinator for this PAN-DBT 1000 virus genome sequence consortium. NIBMG is an institute in Kolkata from DBT Institute. So they are basically coordinating this, and under that we sequence 225 genomes from Odisha. So we thank for their support, help, and encouragement. And finally, our ILS faculties, colleagues, all the students and the staff of ILS, because this is like a big effort that we are doing, testing so much, every day we are doing around 1000 to 1200 samples at ILS, and their inspiring efforts during this COVID-19 pandemic. So at last, I would like to thanks all the organizers again for giving this opportunity to present this walk, and then I hope this will be helpful for understanding the COVID-19 disease. Thanks. So is there any questions later on?
Thank you, Dr. Raghav, for the exemplary talk centered around the topic of COVID-19. It were viruses that emerged as a global pandemic in the current scenario. It was really interesting to find that there are five clades of the reported clades of the virus in our population, that as determined by the phylogenetic analysis of your group. And I must admit the talk was highly informative, not only to the researchers working in this field, but also to the young students who would be highly benefited by this talk. With this, I take up the various questions which are asked by the students as well as the researchers. The first question is asked by Kumar Janmajaya. From which clade the D614G mutant clade has evolved? Excuse me, sir, can you hear me? Dr. Raghav?
Hello.
Hello? Yes, Dr. Raghav, can you hear me?
Is there? Yeah, now I can hear you. Yeah.
The question
There was
was
some
asked by
problem
Kumar.
in between, or what?
No, I can hear you. You can't hear me?
Yeah, no, and it's fine. But I am saying
Yeah.
during presentation there was problem of internet. I don't know because recently.
No, the presentation was good. It was smooth. Okay.
Yes,
So
tell
the
me.
question is
Yeah, the question is asked by the audience, Kumar Janmaja, from
Okay.
which clade the
Yeah, because
D614,
recently,
sorry, from which clade the D614G mutant clade has evolved?
yeah, basically, in the start when 19A19B was there in March, April, there the number of this D614G was really less. But with time, it increased in June or end of May, there were more number. So it basically, if you say in June when we looked at, it was basically 80% of the individual has these mutations test. And in the initial it
Okay.
was 40%
Okay.
in March and
Another
April.
question.
So it shows like it's
Okay.
evolved quite fast, and this mutation has increased in number. So 20A and 20B clade is much more. So and all of them has these mutations now.
Okay. So there is another question from the same Kumar Janmaja. Are there any cases where an asymptomatic patient has died of COVID-19?
Yeah, Niliptam.
Yeah. The question is, are there any cases where an asymptomatic patient has died of COVID-19?
There is a lag, I think, eh?
Yeah, there is, I think, some connectivity issue from your side because I can hear it and see clearly.
It's difficult to say because I'm not from hospital. Uh, I think
Okay, so
yeah,
another
yeah. So
question is
I'm also getting your voice clearly.
okay. So, can you hear the question? Are there any cases where an
So there is some
asymptomatic
lag, it appears, but I'm getting your voice clearly.
okay. Yeah, so you can answer the question if you can hear my voice. The question is, are there any cases where an asymptomatic patient has died of COVID-19? So, is it possible that asymptomatic cases are also susceptible to die of COVID-19? Only severe cases are susceptible for to be dead by this.
It's difficult for me to tell
Okay.
because I'm not a clinician, so we don't.
You don't probably don't have that information that whether asymptomatic cases how many have died. Yes. Okay, so there is another question from Somya Ranjan. uh, how can one vaccine will be effective against 5 clades of SARS-CoV-2?
I I don't. It should be severity because if the layer might be on the similarity of anti. So what you look into antigens that are very similar between different strains, and then you try to
Okay.
develop basically vaccines. Else it will not work for all. But still, we don't know how much efficacious the vaccine will be. We don't know because anyway we see that they develop very less antibodies with normal infection. So let's see. The vaccine will develop how much antibodies. It's still a question
Still a question under
yeah,
investigation, I suppose.
under
Okay, so
investigation.
we have a question from Nisha Patro. Uh,
Yeah.
what exactly happens in the asymptomatic cases?
Yeah, asymptomatic cases, that's why I said, like in context now, what we feel that they have very strong antibodies. I think they have very strong antibodies. Like in context, I showed now, like in context, they have very strong antibodies. So these antibodies most probably are neutralizing the virus continuously. And that's the reason they don't get symptoms.
Okay. Actually, Nisha Patra madam is wife of our honorable vice chancellor, Ishan sir. So, since sir is not infected, so she is probably interested to know that. And this is not
So,
only the
but
question. somehow I think we all are interested to know that what exactly happens in the asymptomatic cases.
No, so that's why I said that it would be good to really check if they have also good amount of antibodies. So it's good to check some of the asymptomatic cases that they don't develop at all any symptoms. It would be good to check
Mhm.
if they have the similar high level of IgA antibodies, because if you have high level of IgA antibodies, it will neutralize very fast in the respiratory tract, the viruses. So you will be positive, but you will not get any symptoms because your virus is neutralized very fast by the body. So it would be good to check if the antibodies are really high.
Okay. Thank you, sir. Now we have another question from one of the faculty from Department of Zoology, Dr. Dhananjay Soren, and his question is: could you please elaborate on specific amino acid mutations like D2G between asymptomatic and symptomatic infection in V20 clades?
Oh, I would say we didn't stratify that, but it would be really good to look into if we can check in asymptomatic versus symptomatic what type of. But as I know, it's not different. It's not depending on this mutation. It's basically depending on the host response. Because if you, as I told you, that 80% of the cases we have this mutation, and asymptomatic is nearly 80%. So definitely it doesn't depend on this mutation. It's basically the host response that really controls that. It's not because of this mutation. Yeah.
So another question is from Swashish Pusti. What is the chance
Mhm.
of getting false negative or false positive for corona by RT-PCR test?
There are always chances, but definitely the RT-PCR is the test of choice, because that is the only way that you can get more than 95% specificity and sensitivity. But still, if the viral load is very low and the sample was not taken properly, the swab is not taken properly, then the viral load might be really low. So it depends on the sample collection also. But in terms of if you have collected nicely sample, there are high chances with RT-PCR you should be detecting it. There is no way that it will not be detected.
Okay.
So it depends on the viral load.
Depends on the viral load. Okay.
Yeah.
Okay. So we have another question from Jyoti Prava. Is there any difference in structure of AC2 receptor, that ACE2 receptor, for different case of the virus?
It's very interesting question, I would say, because it's also important to look into if there is a mutation in ACE2 receptor. It might be really changing the affinity of virus spike protein with the ACE2. I agree. So definitely, if the ACE2 variability is there, it might be that some person will not get high infection. So it would be interesting to look into in terms of host genetics. We have plans to do some host genetics like exome sequencing, but that's a lot of work. So we have not done yet. But that would
So
be
a lot of,
definitely interesting to look into population. Yeah. What is the
yeah.
difference?
Yeah, we're still halfway, I think, in the process of investigating the COVID
Yeah,
virus
yeah,
infection.
yeah.
Yeah.
Actually,
So we have,
before, I can tell one thing, because before people were looking into mostly virus side and then what type of mutation, what type of virus. Now people are looking into more into host responses and all.
I agree.
So let's
Yes.
see what comes out.
So it's not only the virus, but also the host response, which matters a lot, which
That,
which determines
that is the
what
major
exactly.
difference.
Yes, yes. I get.
Yeah, that is the
So we
major
have,
difference. I think host response.
yeah, we have another question from Nisha Patra, ma'am. That can
Yeah.
asymptomatic patients transmit the virus? I think that's the question we all of us are quite excited
Yeah.
to know.
Definitely, it's a open query. That's an interesting question. The reason why we say that asymptomatic cases cannot spread that much, because the reason is you will not sneeze, you will not cough, and
Right,
definitely
right.
there will be less aerosols. And if there are less aerosols, there will be less virus particles. So that's the reason we say that symptomatic cases can spread much more, because asymptomatic will not sneeze or cough in open public. And the
Mhm.
major virus infection will happen because of aerosols. So that is the major reason. That's why later on ICMR had changed that, and WHO has also changed that. that symptomatic cases don't spread virus, and asymptomatic cases basically asymptomatic don't spread, symptomatic spread the virus. That is the reason. Yeah.
So I said by you that aerosol are the major source of transmission. So what kind of mask do you propose that we should wear? Is it only the N95 mask, or the surgical mask will also do the work? I mean, do the job of
Actually,
protecting.
yeah, that's an interesting question because I would suggest if it's an open environment, if you're
Yeah.
outside, even the cloth mask is fine. The reason is it's like a filter, nothing else. But if you're in closed containment, closed room where AC is there, definitely if you have N95, it will help because it's like the aerosol capacity getting inside is very fine, can go inside only. So definitely if you're in closed containment area, like closed room and all, it's better to use N95 or 3-layer mask at least. But outside, I think it's fine. The cloth mask is completely fine. It's like a personal
And
opinion anyway.
they say that the transmission can also occur by the eyes as well. So how do you suggest that we should protect the eyes? Do we need to wear a shield every time, or the mask will do the job? Eyes, we have to wear the specs or something like that to protect our eyes. Because if someone sneezes on our face and we are just wearing the mask, then there are chances that the virus can get inside our body by the eyes. You know. So
Yeah, but
what are your opinions?
this is not like this is an open question still, but I would say from eyes the chances of infection would be quite less. The reason is because nasopharyngeal area, that is the reason where we have more of those epithelial cells, we have ACE2 receptor. But if you have in eyes we have the lacrimal glands and everything in antibodies, they can clear still those virus. But nasopharyngeal area, because we are sucking the air, here we are not sucking anything. Okay, so the chances of getting it inside is less as compared to the nasopharyngeal area because we are taking the air inside. So
I
chances of
know.
infection are really less from air, eyes.
Okay. So recently we are reading about second wave of infection, third wave of infection. So final take, your take on that. That what are these different waves of infection, and when this
Uh,
pandemic is going to end, according to you?
yeah, that's a open-ended question, and we thought that initially that 1 or 2 months it will be done, and we don't need to test again and everything, and now we are still testing. Yeah, so what I would say that definitely still we need to work on that. We checked basically 2 to 5 days antibodies and 8 to 10 days after recovery. We don't see much increase in antibodies. That is surprising. And if you don't have really high amount of antibodies developing with time after infection, then definitely there will be chances of reinfection because you need antibodies and B cell memory cells. Once you get the second infection, there should be already the immune cells to clear it. But if you don't develop those, then there are high chances of reinfection for sure. So that's why people are saying, because they have observed that the antibody levels are not really long time, like residing in infection, like a lot of individuals. So there are chances in those individuals to get reinfection. Maybe what can happen later on, I would like personal opinion again, that it might be that the antibody levels may be checked and B cell and T cell memory levels can be checked for the individuals. And then can we say, okay, this person will not get reinfection, whereas this person can get the reinfection. Those type of tests can be done. That is not a very problematic test to do.
Okay.
You're getting signals
So
from the other side.
yeah. So when do you think that this pandemic is going to end? Any suggestion? When we can expect
In
that we
such a case,
can have a webinar or a seminar?
yes, I hope so. It should be fast because it's not that interactive. If you do give webinar, it's not like that interactive. Uh, I don't know. In India, definitely it will take more time because it appears it will go till November for the peak and all. The numbers are increasing like drastically. So here it will take time, but it will go down. But second wave, it will come. We don't know. So still, it's open. Tomorrow, what will happen? We don't know.
The final question from for today's afternoon, session, we have
Yeah.
from Dr. Kajali Das from OU80. She has the question that does furin activation of spike takes place in the tissue-specific manner?
There are only not furin. Basically, there is TMPRSS2, definitely, because the reason is the expression of this furin like protease or the TMPRSS2 is along with ACE2. So you have the host cell, you have ACE2 receptor and TMPRSS2 are furin proteases on the surface. And as ACE2 receptor expression is different, this protease expression is also different on cells. So it depends on the cleavage. First, the cleavage will happen, then the infection will happen, and then the virus will go inside the cells. So it will vary from tissue to tissue for sure. And recently, that's the reason that people said, like, in gut also, in intestine, you have more of ACE2 receptors. So that's the reason this diarrhea and other symptoms, people said, like, because the virus can infect there, and then you will get those symptoms. So nowadays, any symptom you can say like it's like a COVID-19 symptoms.
Yeah. Okay. Thank you, Dr. Raghav, for sparing time for us from your busy schedule. Now I request our Honorable Vice Chancellor, Professor Ishan Patra, to give the presidential remark. Thank you, sir.
Thank you, Nalita. Thank you.
Well, Sunil, I must say this was a very, very interesting lecture.
Thank you, sir. Thank you.
I'm fortunate to have you in this series, and you have actually set a foundation and a bridge course for the lecture that is coming up on immunity and COVID-19
Uh-huh.
by Dr. Mehra
Doctor,
and Kemp.
Doctor M.
Yeah, it is
Yeah,
there in
yeah,
this.
I know. He's one of the top. So
Uh, it's so
yes, I saw
interesting
it.
that you started the beginning slides. They are really, you are a very good teacher, I must say that. And if there are many teachers sitting over here, they should now note down your name and call you for immunology teaching and genetic teaching to their programs through this webinar thing. I will
I
begin
would be
with
happy to.
the last statement that was given. Like COVID, whatever it did one good thing. It has
Yes.
made us learn this mode of knowing
Online.
and yeah, disseminating knowledge and interacting through the webinar online mode. And we were always postponing this, and now I understand as an education leader. What I understand is this mode of activity should remain, and we have really become very good in this. India, at least, have become very good in this. What I have noted from
Yes.
your lecture is so nicely lined up information in a way that you first introduced us to the structure of the virus, why this virus is different and other viruses, why this one is more dangerous and why it could cause a pandemic, to what kind of what varieties of mutants of the virus are available. And you told us of how their infectivity is either more or less depending upon this. And most interestingly, you gave us the basic information as to what makes this virus uh, dangerous in some cases, and some cases it is not affecting the as much it is feared to be.
Still we are investigating, sir. Let's see.
Yeah, there are lot many things to investigate. I must say this pandemic can go, but studies on COVID will continue. I tell you, because this is going to give us much more information that we have really have now. And what I find
Yes.
is there is now a bifurcation of the study, the symptomatic and asymptomatic clades of this virus and how it happens. And everybody is people are really scared if there is no symptom, and then all of a sudden it starts acting upon the individual, and then you really I do not identify that you are already infested. You do not go for a testing, you do not go for any treatment, and then all of a sudden you see that it has silently, it has become a silent killer, and it has
Yes.
put you to a stage where you are not able to really come out of it. It may cause anything. And as regards eye, as a neuroscientist, I must tell you, this was discussed by Pankaj yesterday. Is touch, if you touch the eye, and particularly through the, um, it can go, again, it has to go to the nasal track only. Your
Yeah.
lecture was fantastic.
Yeah, definitely.
We really,
The percentage would be quite.
really, pardon?
Yeah,
You said
basically
something.
from I still from I we think like
Yeah,
the
it can go
percentage
to
of
the brain
infection.
also. Yeah, yeah, yeah. It can go to the brain also. Yes. So
Yes.
I'm extremely happy to listen to this lecture, and we are all very happy to listen to such a. I mean, it is so difficult for somebody who knows too much to come down and talk to people who do not know anything of it. I say, for example, there are so many things I learned today from your lecture, and it was very nice. I must give you a big thank you for the effort that you made. I must close by saying you are definitely a very good teacher. I will like my
Thank you,
people to
sir.
remember.
Thank you for your encouraging.
This is a very nice lecture. I appreciate. Thank you very much, and I thank my team who are behind the screen to do all this. There are many people who are not seen, and they are doing a lot of
Yes.
work. Thank you very much, and I thank Professor Parida of introducing you to me and asking that you could give this lecture to our people. Thank you very much, Sunil. All the best. Become a very great scientist one day. That's my blessings for you. Thank you so much.
Thank you.
Thank you.
Thank you very much, sir. Thank you for encouraging words. Thanks to all of you who have joined us today. Thank you very much, Raghav.
Thank you all for joining this webinar. Tomorrow, on the fourth day, we are going to have an interesting discussion by Professor Theodore Price from University of Texas at Dallas, and he is a practicing clinician and has a vast experience on chronic pain and pain management. So please do join us tomorrow for this webinar on where Dr. Price will be in conversation with Dr. Murmun Chattopadhyay. So it is a different format altogether. So I invite you all for the tomorrow's webinar. Thank you. Thank you for your attendance and presence.